So DNA double-stranded structure can be changed into a single-strand structure. And when that happens, we call that denaturation of DNA. So think about it. So double-strand structure is natural DNA form, right? Double helical structure. But that can be changed into a single-stranded form. So two strands can be separated out. That's the denaturation we're talking about. How can you do that? So, that double-stranded conformation is made with bunch of hydrogen bonds between the bases, right? Remember, hydrogen bonds are non-covalent bonds. They are relatively weak bonds. So those hydrogen bonds can be broken up if you expose your DNA to the physical conditions that can break up hydrogen bonds. For example, that physical condition could be heat. So high thermal energy can surely break all those covalent bonds, so double-strand DNA can become single-stranded. Okay, heat treatment could do that. So if you look at this graph, okay, so it measures the conversion of double-strand DNA into the single-strand form. So how do you do that? So if you treat your DNA, it's a pain in the butt. Treat your DNA with the high temperature condition, you can measure the conversion because so this is OD260. OD260 is the UV light absorption of your DNA. So if you measure your DNA using a spectrophotometer, and the DNA will absorb a certain amount of light. Okay, so when it's a double-stranded conformation form and the single-strand conformation form, the light absorption pattern will change dramatically. Okay, so from a lower absorption, you will see a higher absorption if double-strand DNA changes form to single-strand DNA conformation. So that's what you see. So this conversion can be made under different treatment conditions. For example, high heat treatment, so up to maybe 60 degrees, they will stay as double-strand form. But if you continually heat up your DNA, they will eventually move to the single-strand form, okay, because of the heat breaking up all those hydrogen bonds, making double-strand into single-strand form. Okay, so there is a conversion made. Okay, so yes, heat is one of the conditions that can denature your DNA. Then how about reversing the process? Can you reverse this? So single-strand DNA becoming the double-strand form, is it possible? The answer is yes. If you remove heat, keep these DNA two-strand into the same space, they can go back to double-strand form. We call that renaturation if that happens. Okay, so we can reverse this by removing the heat. Okay, so single-strand can become double-strand form. So like I just told you, the conversion of DNA conformation can be measured using this machine called spectrophotometer. So inside of this box, there's a light, okay, maybe tungsten with deuterium light that generates UV light. You can select different light wavelength using this prism, okay, and then selected light like 260 nanometer range can be shined through your DNA sample that is dissolved in water, okay, and your double-strand DNA will absorb a certain amount of light, and the light absorption will show up displayed on this machine. That's a spectrophotometer. Okay, so normally at 260 nanometer, if your DNA is double-stranded, a certain amount of DNA will give you the absorption of 1 OD, okay, about 50 microgram per milliliter concentration. Okay, but the same DNA will absorb more light, okay, if they become single-stranded. Okay, if they broke up, all hydrogen bonds become existing as single-stranded, the absorption will become 1.37 OD, although there's no change on the concentration. Okay, and if DNA nucleotides got chopped up and they become free nucleotides, then the absorption will even go up higher. It will become 1.6 OD. So the more, absorption is absorbed if DNA structure is changed. So maybe bases are exposed, then there will become higher absorption at 1.3. And if even the nucleotides are chopped up, then you will get high absorption. That's what you can do with this spectrophotometer. Okay. So when you do an experiment on those melting and denaturation and renaturation, okay, there's an important concept that you need to know. So there's a concept called melting temperature. Okay, melting temperature means it's the temperature at which, okay, at A260 the observance was a half of its fully melted state. Okay, what that means is, you see, remember this graph. Okay, so from here 1 OD, it becomes, okay, 1.3 OD, whatever. So 2 strand and double strand and single strand. Okay, it's the half of that conversion. Okay, the temperature, about 50% of your DNA will be melted. Okay, and then the other 50 is melted, the other 50 is not melted, about here. That temperature will be defined as a melting temperature. Okay, TM. Does it make sense? So here's that the definition of the TM. So the melting point temperature, about 50% of your DNA will become melted. You just as a single strand. So we're talking about that. About 50% of DNA, you just as single stranded. The other 50% you just as double stranded. The temperature that makes you this is melting temperature. Okay. So the melting temperature could vary based on the DNA sequence. That's what it means. Okay, so it depends on what nucleotide you have in your DNA. Okay, if your DNA has more of G and C's, okay, instead of A's and T's, the melting temperature TM will go up higher within a given same amount of DNA. Okay, does it make sense? Okay, if you have 2 DNA molecules made up with maybe 100 nucleotide in each, okay, but one DNA has a lot of G's and C's compared to the other DNA that has a lot of A's and T's. Okay, which DNA will melt it easier? That's the question. Okay, the answer is, of course, the DNA with a lot of A's and T's will melt much quicker at a lower temperature. The reason is, you know, A and T, what number of hydrogen bonds are found? Double, right? Compared to G and C's, there are triple hydrogen bonds found. So, of course, it will take a higher energy to break up triple hydrogen bonds instead of double hydrogen bond. That's the reason. Make sense? And how about, can you treat DNA with salt and would they melt? So they will denature and become single-stranded? The answer is yes. So you can actually do the same experiment against a different salt concentration. So yes. And you can even combine both the salt and maybe heat temperature together. So higher salt concentration of the solution of DNA is dissolved in, the greater the TM. So what does that mean? So I think salt can actually protect DNA from melting. Hydrogen bonds can be protected. Okay, so that's what the scientists found. Okay. So this graph shows you the difference, and the high salt concentration, the TM, okay, conversion, versus low salt concentration, the TM conversion. Okay, so they are different. That's what you see there. Okay, it's a low salt concentration, high salt concentration. The melting temperature is different. Okay. Okay, so now we're going to talk about another term, hybridization. So we learned about denaturation, renaturation. Then what is a hybridization? So think about that renaturation process when 2 single-strand DNA come back together and making a double-strand conformation. That's a renaturation, right? But you can think about the condition where when renaturation happens, but those 2 strands of DNA are not 100% complementary. So their size are not matching up. So their sizes may be different, and then maybe they have maybe not 100% complementary match. Okay, think about those conditions. So there's a dissimilar size between those 2 strands, or the identity of the strands are different. Maybe one is made out of DNA, the other one is RNA, and there may be some mismatch there. Okay, under those conditions, if the renaturation happens, we're going to consider that as a hybridization, because when you form this double-stranded structure, that's a hybrid instead of the double-stranded DNA that's complete, you know, DNA made out of 2 strands that are equal size with 100% base complementarity. Okay, so understand the hybridization difference. Okay, so hybridization could happen something like that, maybe shorter DNA binding with a longer DNA strand, or it could be two DNA came from two different species, so there should be some nucleotide mismatch, but if they bind together forming a double strand structure, we call that a hybrid as well. So that's a hybridization. Make sense? So why is that hybridization important? Think about it. So hybridization can form, like this case, between the DNA coming from two different strands. But the thing is, there is a similarity in the DNA if these two DNA strands may be encoding the same kind of similar gene information. Maybe there's a gene that is very important for the living systems. Okay, maybe it's the gene important for the mitochondrial function. Okay, then that gene may be conserved throughout the evolution. Although there are different species, they may have similar genes. We call those homologous genes. If you compare the sequence of the homologous genes, you can see very high similarities. Okay, so they can be even hybridized into double-strand conformation. So when you compare the DNA sequence, hybridization is a tool to measure the likeness, similarities of the gene sequences. Make sense? And then DNA structure. So we talked about double-strand DNA. So if you look at your DNA, the chromosomal DNA under the natural condition, would they exist as double-strand DNA, kind of relaxed lines? Actually, the answer is not. Okay, no. So yeah, so the DNA structure you're familiar with is the linear structure. So like double strand like this, right? So if you look at some living systems, some eukaryotic cells do have linear DNA, meaning there's one end of the DNA and there's the other end. So it's a line if you stretch out your DNA molecule. Some eukaryotic cells do have linear DNA, like what human chromosomal DNA is linear. There's one end and there's the other end. So there are about 46 linear DNA existing inside of a single cell in the eukaryotic human cells. Okay, but if you look at the prokaryotic DNA, chromosomal DNA molecules, they are not linear. Actually, they are existing as a circular DNA form, just like this. Okay, you cannot find one end and the other end. They are connected together, so it's a circle. Okay, so many eukaryotic DNA, chromosomal DNA is a linear form, but prokaryotic DNA exists as a circular form. Okay, and then viral DNAs are unique. Okay, some of them may be linear, some of them may be circular. Okay, so and then linear can be changed into circular form if they have cohesive ends. Okay, it kind of varies, depends on what living system you're looking at. And the DNA topology. DNA has a flexible structure, but it's a covalently closed DNA structure, and the DNA has extremely long length. And if that's the case, they're subject to topological constraint. Topological constraint means they kind of coil up like this. Instead of using as a relaxed circular line, they coil up more, negatively supercoiled. Okay, so actually the original structure inside of cell is a negative supercoiled structure. Okay, what I mean is this. Instead of existing as circular DNA like this in the bacterial cells, actually that DNA adapts this conformation. It's kind of coiled up again more. So that's a negative supercoiling. So supercoiling happens in your DNA, regardless if it's linear or circular. So you'll find supercoiled DNA chromosomal structure from the cells. If it's a bacterial cell, you'll see something like that. But if it's maybe eukaryotic linear DNA, you will see something like that. So it's a supercoiled, DNA structure that happens in almost all living DNA system. Okay, so beta helix strands are supercoiled further to the opposite direction. So negative supercoiling is happening. That's what you see. Okay.